My name's Steve Long. I'm a professor of crop sciences and plant biology at the University of Illinois. I also have a visiting position in plant sciences at the University of Oxford. So today I want to tell you about how we're actually engineering photosynthesis for greater efficiency in crops. So to get higher yields without necessarily using more water and more nitrogen. So first of all, what I've put in the handout that you have is what you should be able to do after this class. From the information I provide to you and from the associated reading. So basically this is a list of the topics that we'll be working through in this class. And I want to start by just outlining the predicted challenges we have going ahead. Why we're going to need considerably more crop production by the middle of this century. Now, if we look at world food production, actually, directly or indirectly, about 2/3 of our calories come from just 4 crops. So maize, wheat, rice, and soy. Soy comes a very distant fourth. However, we should keep in mind that it is a major protein source. So in fact, it is the largest vegetable protein source. Now, what is the problem? Well, if we look at the production of these crops over the last 50 years, what we see is that there's been almost a linear increase in the amount of rice, the amount of wheat we're producing per hectare of land. So we could say, well, if we can carry on doing that, by 2050 we'll be where these solid lines are on the graph. The problem is the United Nations Food and Agricultural Organization, various other organizations predict that by 2050 we're going to need considerably more food than that. So we can see a growing divide in supply and demand. Now, there's an underlying problem to this that actually if we go into these numbers more deeply, what we see, for example, in the case of rice, and here I've illustrated improvement in rice production in the 3 biggest producers of rice. That's India, Indonesia, and China. And what we see is that in the 70s and 80s, they were improving their productivity per hectare of land by about 30% per decade. And of course, these were the years of the Green Revolution. When we go into the first decade of this century, what we see is that these numbers have declined to single figures. In the case of wheat, actually, we're not seeing any increase at a global level. So, in fact, what this is telling us, it's getting harder and harder to get the increases that we saw over the last 50 years. Now, getting food supply correct is really important, as Marie Antoinette and Louis XVI could tell you if they'd stayed around. Because if we look at disruption of countries in the past 200 years and right up to the current decade, it's often caused by food supply. That has precipitated most of the revolutions that have led to political change in the last 200 years. And if you don't get it right, you can literally lose your head over it. So this is the United Nations Food and Agricultural Organization report on the state of food supply around the world. And what this report shows is that after several years in which food supply has been improving, fewer people in the world are malnourished. This situation has now been reversed, and more people are becoming malnourished year by year, reflecting this separation of supply and demand. Now, in the West, what this means is that if supply begins to fall below demand, we'll be paying more for our food. But probably we barely notice it because really, on average, food is a relatively small amount of our household costs. In the poorest countries of the world, though, it may be 60 or 80% of household income goes on food. So if prices double, that is a catastrophe for those households. And this was recognized by the Bill and Melinda Gates Foundation, who set out to try and solve diseases in the poorest countries in the world. And they realized that much of that disease incidence was caused by the fact that people were malnourished. So they've now also invested heavily in agricultural development. Now, what we can see on this graph is that when there was a decline in particularly wheat supply in the world in around about 2008, 2009, again 2011, what we see then is that prices spike. And although supply might go down 10%, prices can go up 300% because of this difference in supply and demand. So that has a huge impact in the poorest in the world. And many analysts would say the Arab Spring grew out of that. Before we saw changes in the politics in North African countries and countries in the Near East, we had these food riots, and this is from Egypt, the famous bread helmet, where there were riots due to the price of flour for making bread. So as we go forward, these problems could get worse and worse. Now we could solve them in different ways. Reducing demand. If more of us become vegetarian, then fewer calories are needed in terms of primary foodstuffs. We could increase production. We could avoid losses from, for example, storage, supply chains, and so on. However, actually, if we look at what is happening, these things are going in the opposite direction. The global population is becoming more urban. That means that people are not eating directly from their small holdings. The food has to be transported to cities, had to be marketed, etc. That all leads to more losses. And as economies develop, as they are, for example, in China, as people have more money, they want to have more meat, just as we do in the West. And so these things, if anything, are going in the opposite direction, which leaves us with increasing production. And we have the elephant in the room, which is climate change. That is not even taken into account in these future projections. Now, of course, we could get more food by destroying yet more of the world's natural ecosystems and planting those with crops. However, what we have left is often quite poor land, quite poor soils. So there's no guarantee we could get the same amount of food from them that we get from the acres or the hectares we're using today. So we need to achieve more on the land we're already using. And we have another problem. What we were pointing out in this study is that as we manage to increase yields, we also need more water. But there isn't more water to be had. And so we're going to run out of that resource as well. So not only do we need to improve the productivity of our plants, but we need to do it sustainably with the same amount of water and fertilizer that we're using today. So what I want to do now is to explain to you with an equation why photosynthesis, the subject of this lecture, is important in all these equations. So if we look at what will determine the yield we will get on a hectare of land at a given location, assuming that we have enough water, enough nutrients, we defend that crop against pests and diseases, then the yield is determined by the amount of solar radiation, visible solar radiation that drives photosynthesis, the amount of that over the growing season, multiplied by how efficient that crop is at capturing that solar radiation, how efficient it is at converting that intercepted radiation into plant biomass, multiplied by how good it is at partitioning that biomass into the part of the crop we care about. So the grain of rice, the seed of soybean. Now, these are some numbers from a modern soybean cultivar growing on our university farm here in Illinois. And what we can see is that interception efficiency is already a high number. If we look at this one, partitioning efficiency, how much of the biomass ends up in the seed of soybean, 60% is a remarkably high number. What this tells us is there's not really much room for improvement there. However, if we look at this number, conversion efficiency, that's just 0.03, 3%. So it is quite low. Now, what we should do, though, is look at what are the theoretical maxima of these. And so what I've done here is plotted numbers here for our major crops before the Green Revolution against the theoretical maxima. And then what did the Green Revolution do? Well, the Green Revolution hugely increased this partitioning efficiency. Before the Green Revolution, the average for rice cultivars was about 30%. Afterwards, it's 60 to 65%. And if we're still going to have some stem to hold the grain at harvest, then we realize there's probably very little room for any further improvement. Also, interception efficiency improved quite a lot during the Green Revolution. If we look at conversion efficiency, that hardly changed. So today's crops have hardly improved over those of 50, 100 years ago. So what is conversion efficiency? Well, it's photosynthesis. How efficient is photosynthesis at converting absorbed sunlight energy into plant biomass? So why do we think if we could improve photosynthesis, we would actually get more yield? Well, this is another experiment on our farm right next to where I'm standing now. Of course, that's during the summer. And this is soybean. And you can see a ring around that soybean. And we have several of these rings in this experiment. What that ring is doing is elevating carbon dioxide at an even level throughout the year from planting the crop to harvest. Now, carbon dioxide is a limiting substrate for photosynthesis. So if we artificially increase it, which we were doing to try and understand how this crop will perform in the future elevated atmosphere of the globe, if we elevate CO2, we boost photosynthesis. We actually measured that and we boosted photosynthesis by 25%. Does that give us more yield? It does. In soybean, it gives us about 16% more, rice 12%, wheat 15%. So this told us if we could boost photosynthesis in some way, we will get more yield. Now, another demand comes from breeders. And here I had the chance to meet with Longping Yuan, who won the World Food Prize for breeding cultivars of rice with very high yields. And behind us is a picture of one of his rice cultivars. And from the top of the plant, almost down to ground level, you can see these massive panicles that he's bred. But he's complaining to me, he's saying, I can make all these panicles, but I don't have enough photosynthesis to fill the grains. When are you guys going to give me more photosynthesis? So why do we think today we might be able to do that? First of all, photosynthesis is the best known of all plant processes. We know every step in the process. It's also genetically highly conserved. So if we can improve it in one plant, we can probably improve it in all plants. We also have high performance computing, which allows us to simulate the whole of a process such as photosynthesis, so that we can then probe it on the computer to say, well, if we intervene, where might we get the best results? Of the millions of things we could do, what might be the most promising? And finally, crop genetic transformation is becoming increasingly routine, even in the public domain, i.e. in universities. So what I want to do now is to outline to you how limitations in the Calvin cycle have been identified and then actually tested in crop plants. And then we go on to look at some other processes that we've been able to improve in this way. So one of the questions that often comes up is, okay, surely evolution will have already done this. Could evolution have left a free lunch on the table? i.e., aren't plants already optimal? And of course, this is quite a challenging question that we've had to think very hard about. And we've really come up with three arguments against this, which have underpinned a lot of our research. First of all, selection is about survival and fecundity, i.e. genes managing to get themselves into the next generation. Many of you might be familiar with the plant lab rat, Arabidopsis, which is about this size. This is a very successful plant in the wild, yet it's very unproductive. So improving photosynthesis may not be high on the evolutionary agenda. Now, another big change, of course, is that our crop ancestors evolved in a CO2 concentration of about 220 parts per million. That is the average of the last 25 million years. Today, outside, the concentration is almost double that, over 400 parts per million. Half of that increase has occurred in the last 50 years. That's very few generations for natural selection or breeding to act on. So we've changed a limiting substrate of photosynthesis. We've doubled in a very short period of time, and I'd argue that plants are not adapted to that change. If we look at crop ancestors, this is one of the ancestors of wheat. Next to it is a modern crop of wheat. These plants are usually growing in very open habitats, producing very few leaves, so all of those leaves are in sun. Today we grow these in very dense populations where they're producing many layers of leaves. So in fact, most of their leaves are in the shade. So we've taken sun-adapted plants and we're making them shade plants today. And again, a lot of our research suggests they have not adapted to this situation. So that suggests there are opportunities which breeding and natural selection could not have taken advantage of. Well, we actually look at this on a computer. Now, take a car production line. Imagine you've got 1,000 workers. What you want to know is, where do I put my 1,000 workers to maximize the production of cars? Now, you could do that empirically by moving people around all the time. But a better way to do it is to just simulate the whole process on a computer. And a computer could then work through all of the possible permutations in several minutes, rather than the many days it would take you to move all your workers around. And of course, this is the foundation of systems analysis, which is used very heavily in industry. So what we've done is to take the Calvin cycle, the photosynthetic process, represent each one of those as a differential equation, assimilating what we know about the biology of each of those steps. So, for example, using Michaelis-Menten equations for many of the steps in the Calvin cycle. So once we've got this system of differential equations, then we can numerically integrate it to say, okay, with what we have in the plant, how does this behave? Now, I've made this sound very easy, but actually this was about 20 years of work to get this going. And one of the things we found was numerical integration is actually computationally quite difficult. So you might wonder, why have I got a rocket there? Well, one day we were talking about this problem at our computer center, And one of the mathematicians said to me, oh, I have exactly the same problem with balancing rocket motors. So we said, well, can we use your algorithm on our problem to see if it works? And we did that, and it solved our problem. And so that's why on our publication, the middle author is actually a rocket scientist. So don't let anybody tell you that plant biology isn't rocket science. In fact, actually, the mathematician who helped us said, actually, your problem is much more complex than mine. It's mathematically really intriguing, so I want to work with you on this. Now, the problem is that you can do this at a leaf level, but when you're actually growing a crop, a leaf may start off in full sunlight. As the crop grows, it then goes into shade. And over a 24-hour period, those leaves may be in sunlight some of the time, in shade some of the time. So in optimizing the system, you have to not only take account of individual leaves, but how they behave in a whole crop canopy. So we did a lot of mathematical modeling. We came up with many predictions on how we might be able to improve photosynthesis. But of course, models can look great. But when you get down to actually trying to do this in a muddy crop field, things don't look quite so attractive. But fortunately for us, the Bill and Melinda Gates Foundation started reading our publications. And after a discussion with them over several months, they then more or less said, well, how about we put our money where your mouth is, and you show us that this can be done in practice. And so that is what we set about doing. So our approach is, first of all, to start off with the process of photosynthesis. So there are billions of possible changes we could make here. So we used the computational modeling to select out, using optimization routines, what might make the biggest change. Then we go into tobacco, and you might say, why on earth tobacco? Surely the Bill and Melinda Gates Foundation don't want to improve tobacco. And indeed they don't. The reason we use tobacco is, first of all, we can genetically manipulate it very rapidly. And when we've done that, it produces large numbers of seed. So we can then go from genetic change to field trial in 1 year. We couldn't do that with soybean, rice, etc. So what we do is we make the change in tobacco, we test it in the greenhouse. If it looks good there, we go to a replicated field trial. Now, if we get a significant increase in productivity in our field trial, then we go on to the crops that we care about. And that is cowpea, which is the most important vegetable protein source in sub-Saharan Africa, rice, which is the single most important source of calories, direct source of calories for humans. We're also testing these in soybean as well. So this is one example where our optimization routine said, in the Calvin cycle, this enzyme, SBPase, what you have in your plant today is way below what would be optimal. Now, it's not a very big protein, so it ought to be quite easy to increase this. And our colleague in our program, Christine Rains at the University of Essex, upregulated this enzyme in tobacco. And here you can see 3 plants where it's upregulated, 3 controls, and clearly there's a significant productivity increase. So we took that to the field situation and found indeed we did get an increase. Now that led us to question why this is a very small protein. Why hasn't selection just resulted in an increase in this protein? And so I want to show this to you with flux control analysis. So basically, what flux control analysis allows you to do is, if you change, for example, the amount of an enzyme, delta E over E, and you look at the change in the overall process, delta J over J, if your enzyme has a strong control, then what you'll find is that the flux control coefficient will go up a large amount. If it doesn't have control, that number will be close to zero. If we look at experimental data, we can see 4 enzymes where this appears flux control coefficient is high. One of those is SBPAs. Now, why hadn't evolution increased this? Well, what we did was to run the flux control analysis at 3 carbon dioxide concentrations. And what you can see is that 220 parts per million, the average for the last 25 million years, the flux control coefficient for SBPAs is very low. When we go forward to today's concentration, or when we did this, the concentration was a bit lower, we then find that SBPAs is now exerting much more control. And as we go forward to even higher concentrations, it goes up yet more. We also found that temperature would also increase the flux control that this enzyme exerted. We can move forward and say, well, one of the things we could do at Illinois was say, well, we could take those plants and put them in elevated CO2 and see, is this true? If we upregulate this enzyme, do we see a bigger response at higher CO2 than at current CO2? And indeed, that's exactly what we showed with Christine Rains' plants. Now, we also predicted that flux control of this enzyme would be even greater at higher temperature. And so we could test that as well, because we also have within these rings the ability to raise temperature. And what we found is that under elevated CO2 and elevated temperature, the difference between these and control plants was even bigger. So we've found a way to partially future-proof our crop. Now, of course, when you unblock one thing in a production line, a blockage appears somewhere else. And so we could then go back to the model and say, well, we've solved SBPAs, where's the next bottleneck? And indeed, the model then said, well, this enzyme FBP aldolase is the next bottleneck. And so Christine Rains has now added on an additional gene and shown that by doing that, she can get even bigger plants, which we are now testing at Illinois in the field. So one of the other things that we've come up with is the issue of photorespiration. We know that the way plants deal with photorespiration requires a lot of energy. So if Rubisco uses CO2, it goes into the Calvin cycle. If it uses oxygen, it goes into photorespiration. That results in a carbon loss, but it also results in an energy loss. And if we look at what this means for crop production, then the losses due to photorespiration mount up with temperature, because Rubisco discriminates against oxygen more poorly as temperature rises. So photorespiration takes away more and more of productivity as temperatures rise. Now, plants use this very long-winded process of metabolizing glycolate in produced in photorespiration. And that requires a lot of ATP and NADPH. Bacteria and archaea, however, also metabolize glycolate back to glycerate, just as the plant does. But they have found much shorter routes to do this. And there are a number of those metabolic pathways. Now, this had already been tested in Arabidopsis, where Arabidopsis plants proved to be more productive if you put in the bacterial pathway. However, this wasn't replicated in any field experiments. So our colleague in Xingwan Zhu, in this publication, looked theoretically at a number of these pathways. So my colleague here at Illinois and his group, Don Ort, put one of these pathways into tobacco, and in January of 2019, they then published this work showing that by doing this, they could get an incredible 40% increase in the productivity of tobacco. We doubt we would see such a big increase in a food crop, but nevertheless, even if we could get half of that, this would be a major improvement. And they're able to do that without using more water and more nitrogen. So finally, I want to turn to some of the work that's been done in my own lab, and that is actually improving the efficiency with which the plant uses light in sun to shade transitions. Now, when a leaf is in full sunlight, it's receiving far more light than it can use in photosynthesis. But the chlorophyll molecules absorb that light energy. And if that energy is not dissipated in some way, then oxidizing radicals will be produced that would literally bleach the leaf. So evolution has dealt with this by inducing a process which removes that excess energy as heat. Now, when a leaf goes into the shade, the problem is that it will continue dissipating a large proportion of that energy as heat, even though it could now use it all in photosynthesis. And it takes many minutes before we now get to the situation where that heat dissipation is relaxed and that energy can be used in photosynthesis. So essentially what happens is that, of course, when a leaf goes into the shade, photosynthesis drops because there's less light. But it drops far more than it needs to. And it takes many minutes before it recovers to its maximum potential. Now, this is happening all the time in a crop canopy, because as the sun passes across the sky, a point in the leaf, 1 second is in full sunlight, the next second it's in shade of another leaf. And so what we set about doing in our modeling was saying, well, if we take a point in the canopy, and here you can see the second layer down, 1 minute it's in sun, the next minute it's in shade. And this goes on throughout the course of a day. So we then said, well, what does this slow recovery, what does it cost the plant? And what we found was it was about 20 to 30% of potential productivity. So we then worked with colleagues at Berkeley who've been studying the genes involved in this on how could we try and improve it. And we came up with 3 genes that we would need to upregulate to speed this process. And so we put those into tobacco, and then we had 3 different events. And what I'm showing you here is red is showing that the genes we've added to those the plant already has are expressing very strongly. And indeed, what we found physiologically was we had indeed sped up this productivity. And in the greenhouse, these plants next to the wild type look more productive. So what we wanted to know next was, did this work in the field? And indeed, what you can see here is that in replicated field trials, we got about, on average, a 20% increase in productivity. So can we now translate this into crops that count? And of course, you probably know that there's a lot of opposition to genetically modified organisms, so there's a long regulatory process. And although we may have many ways in which we could improve plants, relatively few of those are available to farmers. And I'm showing you here 2 of my heroes, Dennis Gonzalez, who was a plant molecular biologist working with the USDA in Hawaii. And what was happening was the papaya growers there were suffering from ring spot virus, which was destroying their crops. He knew a way where, through genetic modification, he could make those plants resistant to the ring spot virus. So he started growing those plants, showing they were resistant. Then he spent the next 20 years of his life working through the regulatory framework so that those farmers in Hawaii, and they weren't particularly well-off farmers, could actually legally grow these crops and get them to markets. And in fact, he's even got those papayas approved for sale in Japan. Japan is one of the most resistant countries to any genetically modified organisms. And now those papayas are really selling very well in Japan. Those farmers have been able to keep their farms going, their lives going. This is TJ Higgins from Australia, who's worked with the Nigerian government over a long period of time, getting in place a regulatory framework so that genetically modified cowpeas could be made available to crop breeders in Nigeria and also Ghana and Burkina Faso. And so he has just had 2019 approval for his first transgenic cowpea to be marketed in Nigeria. And this is a remarkable breakthrough and really important for smallholder farmers in Nigeria, who are the people who need this technology far more than Western farmers. So we believe that this is a route that we're using for the technology we're developing. I'd also mention that we're also looking at the other side of the equation. What happens when a leaf goes from shade to sun? And again, we've recently analysed this and we found that if we could speed that up, we might be able to get another 20%. So in our Gates Foundation project, we're also working on that. Now, you might say, well, surely we have plenty of food today. Do we really need this? I'd say for the poorest people in the world, we need it today. But if we go forward to 2050, there is a risk that many more people may need it. So, if you think, if we've got an improvement today, it's going to take 20 years before it's in farmers' fields at scale. So we do need to be working on this problem urgently today. Now there may be other solutions. We might all become vegetarian. And this technology will still be very useful because we're really cultivating more land today than we should be. So if we can produce more on land, we can actually take some land out of agricultural production, let it go back to natural ecosystems. So either way, I'd argue these are technologies that society needs. And so maybe we'll need it, maybe we won't. But if we don't develop these technologies today, it may not be there when we really need it. This is the reading list. And so this top one that we published in Cell in 2015 is the one I'd ask you to read to follow through on today's class. I'm giving the other references because these are the sources of the information that I presented to you today. Finally, I'd like to tell you about the internship that we've developed. It's funded by the Bill and Melinda Gates Foundation and the Newton Abrams Fund at the University of Oxford. Last year, 10 students from this class actually joined us in Illinois for the summer to work on different projects, to do practical work in the field, in the laboratory, on improving photosynthesis in crop plants. And I think, if you go to the website, you see what those students said about their experiences. And we hope some of you will be interested in applying to us to join us this summer at the University of Illinois for an immersion experience in crop science research. So Thank you for your attention. And if you want to find out more, we have Twitter, we have Right web account, and also my lab account as well, longlab@illinois.edu. I look forward to hearing from some of you. You have my email.